Welcome, ladies and gentlemen, to my last video of the night. I'm going to try and do this as fast as possible because I have 60 questions to go over. If you've seen my videos before, you know that there's a point in the middle where I kind of go fast because we've discussed so many of the same topic that once we go through the same questions over and over again, it's like boom, 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 like yes, we got it, and the questions become so much easier. So that being said, let me not waste your time and go straight to it. This video is the request that you gave to me in the comment section of my previous video, and I asked you, what do you want me to specifically talk about? So I'm going to talk about everything you requested. I chose the best questions that pertain to what you asked for from previous Regents exams, and a couple of you wanted labs, the 4 mandated labs. I already have a video specifically just on that. It's called Living Environment Part D Lab Section. So go ahead into my channel and look for Part D Lab Section. That is all about the 4 mandated labs: diffusion through a membrane, making connections, beaks of finch, relationships, and biodiversity. I don't have time to make another video. This is my last one. So are you ready to get beyond a 65 where you're going to probably get an 85 or higher? Good luck. Watch this entire video. Watch my videos again in the morning. Wake up refreshed. Let's get to it. Number 1. So one of you asked for cellular respiration, also known as just respiration, but I want to talk about both because both pertain to organisms, cells making their own energy. I want to talk individually about photosynthesis. Now remember, everything that I talk about is information that could end up in a multiple choice question or a short response. So we're going to spend a lot of time on this question number 1 only because it's going to go beyond what you need to know, but how to answer the question where you maximize your points. Check this out. This number has 1, 2, 3, 4, 5, 6 bullet points. That means that this is worth 6 points. And the Regents doesn't give you much lines to work with, so you're going to end up writing things all on the side if you're just not careful and go straight to the answer. So I'm going to talk about that a little later. Photosynthesis is a way for plants to get energy from the sun, and they are autotrophs. Plants are autotrophs. Because autotrophs make their own plants make their own food. I've upgraded, guys. I don't need my digital pet anymore. Plants make their own food. And where does their energy come from? Where does all energy originate? From the sun. So the circle of life, as Elton John describes it, energy starts with the sun, our star. The plants, autotrophs, take the energy from the sun and make glucose. So plants use the process of photosynthesis to make glucose for themselves. Because plants can't just take their roots out of the ground and say, hey, I'm going to walk over there and get me a fly for those Venus flytraps. They are rooted and they stay in the soil. They don't walk around. This isn't Lord of the Rings where the trees start walking around because they have a mind of their own. Plants need to get food for themselves using the process of photosynthesis. So there is a special formula for photosynthesis to happen. So back to my digital pen, because no, I don't need my digital pen. I could do the word equation. So if you're wondering what are the chemical equations or the chemical formulas, go to my previous videos and you will check out what these molecules and these compounds look like in the chemical form. But right now, for time purposes, I'm just going to keep going. So the photosynthesis formula, what plants need, which is also called raw materials. Raw materials for photosynthesis is It needs carbon dioxide. Plants need carbon dioxide. This is going to come back later. Plants take away pollution. Carbon dioxide is the reason why there's global warming. So plants take away carbon dioxide because they need it for photosynthesis. What do plants also need to live? They need water. And then, besides that, we know that on top of this arrow, if you're ever asked, which is rare, it should be energy from the sun. Energy from the sun is also, so plants need carbon dioxide, water, and sunlight. And then what goes on the product side? Think about the entire point of photosynthesis. The entire point of photosynthesis is so that the plant can make glucose. And this is a sugar, also known as a carbohydrate, as you know from my previous videos in the chemistry of life. So plants produce something else for us in the process of photosynthesis, something that we need, and that is oxygen. So all plants that photosynthesize, because it's not just plants, algae, which is a one-celled organism, also photosynthesize. So we have oxygen in our atmosphere because of photosynthesis. So again, these are raw materials. Sun energy, energy from the sun, that is not a raw material. So don't go putting the sun when they're asking for raw materials. No, 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 no. And glucose is what the plant is making for itself, so that it can then do the process of respiration. So only plants and some one-celled organisms do photosynthesis. But all cells, including plants and animal cells, go through the process of respiration, cellular respiration, okay? So I'm going to delete this, and you can always go back if I can do it. Okay, let's see if I could do this. Okay, I'm going to pause it real quick. Okay, so now we're going to talk about cellular respiration, okay? We're going to talk about cellular respiration. Now, let me just fix this a little better. I'll get faster with my annotations, but this is also called, I'm going to start higher here. This is called cellular respiration. What happens in cellular respiration? This is when cells make energy. If you saw my previous videos, you know that the energy is called ATP, ATP energy, also known as adenosine triphosphate. Did I spell this right? Hey, it happens to the best of us. Triphosphate. I want to make sure I spell this right for you. Triphosphate. We'll get it. We'll get it. All you need to know is ATP. That is the name of the chemical molecule that is made through the process of cellular respiration. I'm going to repeat. Cellular respiration is when all living cells, all kinds of cells, plants, animals, all kinds of cells, make energy in the form of ATP. I'm getting excited here because we're getting closer and closer. So let's keep talking about cellular respiration. So cellular respiration, you know what? Let's talk about the formula. The formula of cellular respiration is the opposite of photosynthesis. So think about what you need to live. What do you need to survive? What do you need to live to continue making energy for yourself? Well, you need food. In this case, for the living environment, they want you to think about glucose. So you need a carbohydrate, glucose. You need to breathe. Your cells need to take in oxygen to take it to your cells so that your cells can make ATP energy. So you also need oxygen. Where do you get oxygen from? From wonderful photosynthetic organisms that are giving oxygen to the atmosphere. On top of this line, so these are the raw materials for cellular respiration. On top of this line, enzymes would be put here. I don't think you'll be asked to do that, maybe in a multiple choice, but that is high level. So what is the product after this? After the arrow is the product side. These are the raw materials. You can say the ingredients to make ATP energy. So that's what we're making, ATP. But there are two other things that are made that are waste products. And these waste products are called byproducts as well. This, you also make water and carbon dioxide, which is CO2. We know that this is H2O, this is CO2, glucose is C6H12O6, C being carbon, H being hydrogen, O being oxygen. So glucose plus oxygen, raw materials, gives us this wonderful ATP energy. The byproducts that are a waste that we need to remove is water and carbon dioxide. We remove carbon dioxide by our respiratory system. That's what we breathe out, ladies and gentlemen. We breathe out carbon dioxide, and that's how we get rid of that excess. And also, in our breath, we have water. That's why you can fog up a window or a glass when you go on it. When you breathe on glass or you breathe on a mirror, and it fogs up, those are water molecules. You're breathing it out. Now that we have the background information, let's go and answer number 1 in 2 ways. Yep, we're going to answer it with photosynthesis, and we're also going to answer it with cellular respiration. We're going to do it 2 different ways. All right, guys, here we go. The meat and potatoes. So at this point, if you already knew about photosynthesis and cellular respiration, this is the part you kind of want to skip to. I'm going to do a combination of how to answer this question and how to answer this question so that teachers can find it easy to test, to grade you and give you maximum points, and also how to properly answer these questions and giving the right type of information. You see how there are bullet points here? You got to answer in bullet points as well. Each one of these bullet points is a question. So if you only answer 2 of all these bullet points, you're only going to get those 2 points if you're correct. Also, don't leave blanks. If you leave blanks, you're more likely to not get as many points as you deserve. And remember that living environment, hey, if it makes scientific sense, you can probably get the point. So a lot of these questions can be answered with logic, but some of these questions are actual straight knowledge of biology from class, from what you've learned. So let's get started. Bullet number 1, which you should write in pen, not pencil. They will call you and have you come back home to redo your test in pen, or your test will be invalidated. So here we go. First bullet point, we're choosing photosynthesis. Identify the process being discussed. Wow. What an easy point. If you don't do that, we have problems here. I chose photosynthesis because it says to discuss 1, so we're doing photosynthesis first. So I wrote it. Next bullet point, next question, come on, give me all the points. Identify the organelle where this process occurs. This photosynthesis in the plant cell happens in the chloroplast, not chlorophyll. Not chlorophyll. Chlorophyll is the green pigment in chloroplast, and it's why plants look green, because they photosynthesize. This is the chemical that's used for that solar energy capturing molecule. But photosynthesis happens in the chloroplast. Next, let's go. Let's do this. Identify 2 raw materials. Look at that. Raw materials necessary for this process. I'm getting excited, guys, and I just had coffee. So identify 2 raw materials. The raw materials that is needed, the ingredients for photosynthesis, we'll name 2. Remember, it's not the sun. Do not say the sun. So raw materials is carbon dioxide and water. That's it. No sun. Moving on. Next one. Identify 1 energy-rich molecule that is produced by this process. What is the whole point of photosynthesis? To make glucose, sugar, carbohydrates. That is the energy that the plant needs. So The answer for the fourth one is glucose. Don't just say glucose. Say glucose is produced as an energy-rich molecule. Some teachers might not take your glucose because they might say, wait a minute, which bullet point are they answering? Especially if you start mixing answers around, answer in order. Answer in order, because if you do something like this where you just put glucose, they might not give you the point because they're like, is she or he or she answering the first bullet point? So they want to make sure, number them. A lot of the questions say, oh, numbers 57 to 67. So this will be 57, 58, 59. So number them. Be very clear, guys. Next bullet point. See, even I have to keep track. This is the 1, 2, 3, 4, 5th one. 1, 2, 3, 4, 5th. State how organisms use the energy-rich molecule that is produced. Glucose is used to make ATP energy. Next, because plants get energy from the sun to produce glucose so that they can go through cellular respiration to make their ATP energy. Next, last one, state how a gas produced by this process is recycled in nature. What gas is produced by this process? The gas that's produced in this process is oxygen. Oxygen is produced. You can leave it like that. Oxygen is produced. Look how you didn't need to write that much. This took us, what, 4 minutes? Come on now. And this information doesn't change. It's the same information. I don't have to explain much. Moving on. Oh, no, not moving on. We got to do now cellular respiration. Okay, let's go. Cellular respiration now. So cellular respiration, let's start answering the bullet points. Identify the process being discussed. Easy point. Put your bullet point. When I'm doing this, putting the star, that means you with a pencil can put a little star or you can put a little bullet point. Remember that. If the question's in bullet points, answer in bullet points. Write in pen. This is not pencil. Some teachers do not remind students of that, and then you do the whole test in pencil. Then they have to call you back to come back home. Identify the process. This time we're doing respiration. Next bullet point. Identify the organelle where this process happens. Respiration is to make ATP energy. Where does this happen? In the mitochondria. Next. Identify 2 raw materials necessary for this process. So the raw materials for cellular respiration, to make cellular respiration happen, is glucose and oxygen. But do a complete sentence. Are the raw materials. Materials. Next bullet point, and let me just increase this. Next bullet point. 4th, identify one energy-rich molecule that is produced. Hello, the best energy of them all, ATP, is the energy-rich molecule. Next one. State how organisms use the energy-rich molecule that is produced. State how organisms use, and let me correct this. ATP is used for life processes because we can't live without ATP. Next one. State how a gas produced by this process is recycled in nature. Well, this produces this. Let's just keeping it simple, right? Carbon dioxide is produced, and what needs carbon dioxide? Photosynthesis. For the process of photosynthesis. Done, done, done. Oof. Yes. Let's continue. What do we have next? Let me just minimize this a bit. So here we have a different set of questions. Remember what I've said in the past, that you need to start from the top and work your way to the bottom. These are the most important 3 hours of your life. Of the year. Let's not be too dramatic here. The most important 3 hours of all the work you've done in the year. Take your time, read directions. Base your answer to the questions questions 2 and 3 on the information diagram below. We're going to read this. A student, okay, so this is where we're doing experimental design. Some of you asked if I can do the scientific method. In the living environment regions, it's not necessarily called the scientific method. It's called experimental design. Experimental design is what you need to know, like what characteristics are found in an experiment. So you have to know what a hypothesis is. A hypothesis is never a question. It is a statement. And I'm going to emphasize that in a minute. You need to know what is a dependent variable and an independent variable and a control. Let's see. You need to know what an independent variable is. You need to know what an independent variable is as well. So I chose these questions to help us. This, I have to be honest with you, you got to memorize it. You got to remember what an independent variable is and what a dependent variable is, and how to answer a hypothesis question. Now, let's get started. Let's read, and I will continue to break things down. A student wanted to test the hypothesis that rooting hormones will stimulate the production of new roots at a faster rate than would take place without rooting hormones. 2 stem cuttings of equal length, similar to the ones shown below, were taken from a rose, a begonia, and a geranium plant. The cut end of one cutting from each plant was dipped into the hormone and then planted in wet sand. The other cutting from each plant was planted in wet sand without dipping it into the hormone. I am going to highlight this with a different fun, okay, forget it. But I'm going to highlight this. The other cutting without dipping. There you go. Sorry. I was just making sure that I was highlighting the right thing. This, my friends, is called a control group. A control group is a set of a separate experiment. So you see how they're testing with hormones? A control group is the same experiment without the hormones. It's to see how the plants would act with no treatment, how it would act normally with nothing added to it. That is the control group, because you want to do a comparison. If you just do the hormones, putting the hormones in the plant, you have nothing to compare it to see if it's working. So the control group is the same experiment but no treatment, and you're going to find out what a treatment is called in a minute. Okay, all cuttings were maintained in identical environment conditions. Good. At the end of 4 weeks, all cuttings were removed from the sand and the lengths of the roots that had developed were measured. The results are summarized in the data table below. So let's see what happened. So here we have the measurements of treated with hormone, and here we have the measurements of untreated. So we can see that for the rose there was no effect. For the geranium there was a little bit of an effect. The hormone did have a slight increase in growth as opposed to no hormone. And the begonia grew significantly at 0.5, and untreated it only grew 1 centimeter. This is how I know it's centimeters, this right here. It says the unit. And with the hormone it worked. So the hormone did have an effect on the growth. But let's see if that's even what they're asking for. They love all of this to ask you 2 questions that you could have gotten without reading everything. But listen, read everything. Annotate, highlight, okay? Question, finally, meat and potatoes here. Describe one way the student could make the experiment more valid. In order for you to make the experiment more valid, it has to be done over and over again. Experiment has to be done again and have the same results in each trial. That's it. That's how you make an experiment valid. You do the experiment over and over again, and the results have to be the same. Next question. What purpose did the untreated cutting serve in this experiment? It served as the control group. Period. Moving on. Yes, you got that. You're not going to forget that. I know you're not. You have good memory, and you're going to wake up, sleep well, have good breakfast, right? Okay, base number 4. Not even part of this question anymore. Moving on to something else. Base your answer to the following question on the investigation described below and on your knowledge of biology. This one is going to ask about the independent variable in an experiment. Okay, let's read. As a part of an investigation, 10 beet seedlings in one setup were grown in the dark, while 10 seedlings in another setup were grown in sunlight. All other growth conditions were kept the same in both settings. This is very important, guys. In order for you to do experiment, a control group and an experimental group, you have to keep the conditions the same. So in this case, it's growing seeds, right? The pots have to be the same size, using the same soil, using the same temperature, same room temperature, same seeds. Don't change the seeds, okay? Same exact conditions. The seedlings grown in the dark were white with long, slender stems. These seedlings soon died. The seedlings grown in the sunlight were green and healthy. Identify the independent variable in this investigation. So the independent variable is what is the treatment. So it means the treatment. Treatment being not how you treat the plants. If you treated it soft, if you were gentle with it, no. What did you do to the experiment? What are you testing? How did you treat it? Did you give it a chemical? Did you put a hormone? Did you change the temperature? What was your treatment? In this particular situation, in this particular experiment, the treatment was the light source. You see how in the treatment they had 2 different setups? One grew in the dark and one grew in the sunlight. That was the treatment. So let's just read seedlings. Another setup were grown in the sunlight, all grown the same. Yeah. So the treatment was the light source. One had no light, the other had sunlight. That is the treatment of the experiment, the independent variable. Let's try another one. For the, let me go back to the comment. Independent variable. We're still at independent variable. So Daphnia, these are the cutest little microscopic organisms that are found in pond water. They're so cute. YouTube Daphnia playing the piano. It's not literally playing the piano, but it's just the way they move. It looks like it is. It's super cute. Daphnia are freshwater organisms, sometimes referred to as water fleas. Design an experiment that could be used to test the effects of temperature on the size of Daphnia population. In your experiment design, be sure to identify the independent variable in the experiment. What are we treating the fleas with? What are we going to do to the fleas to see if they reproduce differently? The temperature. So the independent variable, which is the treatment, independent variable is the temperature. We are manipulating the temperature. We are treating it with different temperatures to see if it grows, if they reproduce, if they make more Daphnia. Just a little review on what I said before about the control group and non-control group. The control group in this experiment is Daphnia growing in their natural temperature where they would normally live. The experimental group is Daphnia that grow, let's see, the effects of temperature, that grow in hot temperature, hotter, warmer temperature. Another experimental group, number 2, Daphnia that grow in cold temperature. So this experiment can have 3 groups: the control group, experimental group 1, experimental group 2. Independent variable treatment. Next. Now let's talk about the independent variable. The independent variable, the dependent variable is what can be measured in the experiment. Just giving you a heads up, once we're done with this, I'm going to stop the recording, because if I make the recording an hour long again, it's going to take about an hour to upload. Maybe you guys in the comment section can let me know how my YouTube videos, my very long YouTube videos, like an hour plus, take a short amount of time. I have really good Wi-Fi. I have amazing memory. Everything I use, I save everything Google Drive. So any techies out there, my videos take forever. So I'm gonna make this in shorter spurts so that you can have it as I move along. Dependent variable, what can be measured in the experiment? And what you're measuring is what you're trying to learn about the experiment, your results. So let's read. Start from the top. Base your answer to the following questions on the information below and on your knowledge of biology. A scientist conducted an experiment to test the hypothesis that maple seeds exposed to acid rain will take longer to germinate. Germinate means to grow, than seeds exposed to normal rain, which has a pH of 5.6. The scientists set up 4 groups, each containing 200 maple seeds. The water used for each group had a different pH value: 5.6, 4.0, 3.0, and 2. Very acidic over here. All other conditions were kept the same. After 10 days, the number of seeds that had germinated in each group was counted. Identify the dependent variable. Well, what can we measure here? So after 10 days, the number of seeds that had germinated, so that's the dependent variable. Remember, the dependent variable is what can be measured in the experiment. We're trying to see how many seeds grow depending on these different pHs. So our result that we can measure is the number of seeds. So the dependent variable for this experiment is the number of seeds that had germinated in each group. Done. Period. Let's keep going. One more dependent variable example. We want to do at least 2 for each thing. Okay, start from the top. Base your answer to the following question on the passage below. Some pancetta plants, I think I said it right, have green leaves that turn red. A garden club decided to study the color of the pancetta plants. Panacea. Okay, knowing that this plant changes colors during the short daylight periods of winter, they decided to investigate the effect of different daylight lengths on color change. Design a control experiment using 3 experimental groups that could be used to determine if the number of hours of daytime has an effect on the color change of this plant. But I want to focus in on the dependent variable. This you can figure out. This you can do different control group, experimental groups. I can do that in a minute, but I want to focus on the dependent variable, which is what can be measured. So what can be measured here in this particular experiment? Let's see. Okay, let me make sure. Oh, the dependent variable is the color change of the poinsettias. I think I said this right. Period. That's what we're measuring. We're doing all these different, okay, so let's read it one more time. Some pancetta plants have green leaves that turn red. A garden club decided to study the color change of pancetta plants. Knowing that pancetta change color during the short daylight periods of winter, they decided to investigate the effect of different daylight lengths. Okay, so let's answer the top one. Design a controlled experiment using 3 experimental groups that could be used to determine if the number of hours of daylight has an effect on the color. Oh gosh, this is so easy. Okay, so experimental group number 1, poinsettias control group, growing with normal hours of daylight. Control, normal, regular. Experimental group number 2, the plants growing in only 5 hours of daylight. Experimental group number 3, poinsettias growing in only 10 hours of daylight. Final results, dependent variable measured should be the different types of colors in the pancettas. That's it. Easy peasy. I trust that you won't forget. Okay guys, I'm going to pause the video here so I can upload these 38 minutes, and then we are going to talk about genetics, DNA, protein synthesis, all of that jazz.